Using transects is a valuable method in ecological studies to analyze the distribution of organisms across a specific area. This technique involves laying down a line, known as a transect, across the habitat you wish to study. The primary purpose of using a transect is to understand how the presence and abundance of organisms change across an environmental gradient. This method is particularly useful when studying areas where conditions such as soil type, light availability, moisture levels, or human impact vary significantly across the landscape. A transect can be created using a long measuring tape, rope, or even a marked string, depending on the length and precision required for the study. The transect is laid out in a straight line across the area of interest, crossing different types of habitats or environmental conditions. This line represents a sampling axis, along which data is collected at regular intervals. The point or line intercept method can be used depending on whether you are recording the presence of organisms at specific points along the transect or along its entire length. Quadrats, which are square frames of a standard size, are often used in conjunction with transects to collect more detailed data about the organisms present at each interval. Quadrats are placed at regular intervals along the transect line, and researchers then examine the contents within each quadrat to record the species present and other relevant data. This method allows for the analysis of species distribution and abundance across the environmental gradient covered by the transect. To estimate the percentage cover of a species within a quadrat, the researcher visually assesses how much of the quadrat's area is occupied by that species. This can be done by dividing the quadrat into smaller, equally-sized squares, often by using a grid overlay, making it easier to estimate the percentage cover more accurately. For instance, if a species covers half of the quadrat, it would be recorded as having 50% cover. This technique can be particularly useful when studying plants or sessile animals like barnacles, where the concept of individual organisms is less clear-cut than in mobile species. Once data from multiple quadrats along the transect are collected, it can be used to identify patterns in species distribution, such as which species are dominant in particular areas, how biodiversity changes along the transect, and how different environmental factors might influence these patterns. This information is crucial for understanding ecological relationships and can inform conservation strategies or the management of natural resources. In summary, transects, combined with quadrat sampling, offer a systematic approach to studying the distribution of organisms across varied environments. By estimating the percentage cover within quadrats, researchers gain insight into the relative abundance of species, which can reveal important ecological patterns and inform environmental management practices.